Well, hello, my name's Katie Adolphus and I'm a PhD student in psychology at the Human Appetite Research Unit at the University of Leeds, and I'm also a physiologist by background. And today I'm actually going to talk to you about a topic that interested me while I was studying physiology, and that's about the anatomy and physiology of the eye, and more specifically the process of phototransduction, which is how light energy is converted into electrical nerve impulses which are then transmitted to the brain. And so there's four main parts to my talk today, and some of them require a bit more background knowledge than others, so it might be that you need to go away and do some background reading before you go on to some of the stages in the talk. So firstly, I'm going to cover the anatomy of the eye, so the overall structure and function of the eye and the different components and what they do. I'm then going to talk about the journey through the human eye, so the different processes and structures that light has to go through in order to reach the light sensitive part of the eye, which is at the back of the eye, the retina. I'm then going to discuss the retina in a bit more detail and talk about the light sensitive cells, which are the rod and cone cells. And then finally, I'm going to talk to you about the process of phototransduction, which requires a bit more background knowledge. So first of all, just to start off with, I'm going to talk about the anatomy of the eye, so the different components of the eye and their function. So here is a picture of the human eye, and as you can see, it's quite a complex organ with many different structures, and they kind of orchestrate together in order to produce our vision. And the first structures that I'm going to talk about are the cornea, the iris and the aqueous humor. So the cornea is positioned just there at the front of the eye, and it's the outermost surface of the eye, and it's a transparent structure, and it lets light passes into the eye, and it also helps focus the light on the retina. So it refracts the light, it bends the light and helps it focus on the retina. Then we have the aqueous humor, which is a transparent fluid that's located in the anterior chamber, which is just between the cornea and the lens, and it's produced by the ciliary body. And this fluid provides the nutrients and the oxygen to the cornea. Then we have the iris, which gives the eye our colour, and it's essentially a ring of muscle fibres that controls the diameter of the pupil, so it regulates the amount of light that's let into the eye. So when we're in really bright light, the muscle fibres of the iris contract and it constricts the pupils, and so less light is let into the eye so our eyes don't get damaged. And then in very dim light, a different set of muscle fibres in the iris actually radiate outwards, and they dilate our pupils and lets more light in, so we'll be able to see in more dim and poor light. So then we have the pupil, the lens and the ciliary body. So the pupil is essentially a black hole, and it just lets light pass into the eye, into the retina. And then we have the lens and the ciliary body, and the lens is a transparent, adjustable shape that's positioned by the ciliary body, and it helps focus and bend the light in order to focus it on the retina. So it's the second point that light is focused on the retina, and it's regulated by the ciliary body. So the ciliary body controls the shape of the lens. And then we go on to sort of the main body of the eye. So we have the vitreous humor, which is a gel-like transparent fluid that fills the main body of the eye, so just between the lens and the retina. And then we have the retina, which is the innermost surface of the eye, and it lines the whole of the inner surface of the eye, and this is a layer of nerve cells and photoreceptor cells, which are the light sensing cells. So it's a really important structure in terms of phototransduction. And then we have the optic nerve, which contains over 1 million nerve fibres that are transmitted to the brain. And what's interesting about how the optic nerve is arranged is that all the information from our left eye goes to the right side of our brain, and all the information from our right eye goes to the left side of the brain. So the optic nerve actually crosses at the optic chiasm. And then we have the fovea, which has a high concentration of photoreceptor cells, the cone cells. So it's the point of highest visual acuity in the human eye. We have the choroid, which is the middle layer of the eye, which is essentially a network of blood vessels that supplies the eye with the nutrients and the oxygen. And then we have the sclera, which is the outermost surface of the eye, and it's a durable, tough, thick membrane that protects the whole of the eye. So moving on, now we'll talk about the journey through the human eye, so the different structures that light has to go through in order to reach the retina. So In order for photons of light to pass through the eye, all the structures need to be transparent. They need to be clear for them to pass through onto the retina. And the first structure that light passes through is the cornea, so the transparent structure at the front of the eye. And this protects the eye, it's clear, and it's also where light is initially refracted, and so it's focused on the retina. And what's quite interesting about the cornea, it actually has no blood vessels, and it gets its oxygen directly from the air from a process called diffusion. Then light reaches the aqueous humour, so the transparent fluid-filled compartment in the anterior chamber, and then it goes through the pupil, which is the black hole. And it's the reason why it's black is because most of the light is absorbed by the tissue surrounding the eye, surrounding the pupil. And the pupil regulates how much light can enter the eye by the smooth muscle surrounding the eye in the iris. So then we have the lens, and this is a really important structure in terms of the focusing of light on the retina. And it's located between the aqueous humour and the vitreous humour, and it's a transparent, flexible, biconvex structure. And it refracts, bends the light to help focus on the retina, and it also has a very extensive cell skeleton, and this maintains the shape of the lens. And so the lens is adjustable. It can change shape depending on where the objects we're looking at are positioned. And the ciliary body controls the shape of the lens, and the ciliary body is essentially muscle fibres that just hold the lens in place and controls the shape of the lens. So when we're looking at distant objects, the rays of light are parallel, so the lens remains quite flat and thin in order to focus the light on the retina. But when we look at objects close up, the light rays are more scattered, so the lens needs to contract and become shorter, fatter, more rounded in order to focus the image on the retina. And this process is called accommodation. After the lens, the light reaches the vitreous humour, the gel-like substance that fills the main body of the eye. And this gel-like substance is produced by the retinal cells, and it kind of maintains the shape of the eye and gives the eye its main body. And it also lets light pass through onto the retina, which lines the back of the eye. Then light finally reaches the retina. And the retina contains the light-sensing photoreceptor cells that generate the electrical nerve impulses from the light energy. And the retina is essentially the eye's photographic plate, and it lines the inner surface of the eye, and it contains the photoreceptor cells and also other important nerve cells that signal to the brain. So it has a similar function to the film in a camera. So let's discuss the retina in a bit more detail and look at the different nerve cells that are in the retina. So here is a kind of schematic diagram of the retina, and you can see it's a layered structure. So it contains several layers of nerve cells. These nerve cells convert light energy into electrical signals. And there's three main layers of nerve cells, and they're actually in a counterintuitive arrangement. So light needs to pass through the different layers of nerve cells before it actually reaches the photoreceptor cells. So it's kind of positioned the wrong way round almost. And there's three main layers. So the first layer of the retina we have is the outer nuclear layer, which contains the photoreceptor cells, so the light-sensing cells, and these are the rod and the cone cells. The next layer is the inner nuclear layer, which contains the bipolar cells and the amacrine cells. And the final layer of the retina is the ganglion cell layer, and these contains the ganglion cells, and the ganglion cell axons form part of the optic nerve, which is then transmitted to the brain, to the visual cortex. So if we just look at the photoreceptor cell layer in a bit more detail. So there are two different types of photoreceptor cells. We have rod cells and cone cells, and they're quite similar in structure, but they differ in terms of their roles for light sensing. The first photoreceptor cell we're going to talk about are the rod cells. And these are highly sensitive photoreceptor cells. They can actually respond to just one photon of light, so they're really, really sensitive. And they give us our night vision. So because they can respond to just one photon of light, they can actually function in darkness and very dim light. And they're rod-shaped, this is why they're called rod cells, and they're concentrated in the outer layer, the kind of outer periphery of the retina, so they're more responsible for our peripheral vision. So in the outer segment we have discs of rhodopsin, and rhodopsin is the visual pigment, the light sensing molecule. And rhodopsin is made up of two molecules. It's made up of retinal and a protein called opsin. And retinal is actually derived from vitamin A. We then have the inner segment, which contains the mitochondria, so it supplies the energy to maintain the high metabolic demands of the cell. We then have the cell body with the nucleus. We have the axon, in which the nerve impulse travels down, and then finally the synaptic terminal. The synaptic terminal releases the excitatory neurotransmitter glutamate. How rod cells are actually arranged within the retina is that multiple rod cells actually converge onto the bipolar cells. So this aids in amplifying the signal, the nerve signal that's transmitted down onto the bipolar cells, but it compromises the visual acuity. So because multiple rod cells converge onto multiple bipolar cells, it actually reduces their visual acuity. So the other type of photoreceptor cell is the cone cell, and these function better in bright light, so they need a lot of photons in order for them to become activated. They're more concentrated in the fovea, so the point of highest visual acuity in the eye, and they also give us our colour vision. And again, they have an outer segment which contains the visual pigment, but this time it doesn't contain rhodopsin. They contain 3 different types of opsins that respond to different wavelengths of colour, so red colour, green and blue. We then have the inner segment, which has the mitochondria, which provides the energy for the cell. We also have the cell body with the nucleus, and then the axon, which the nerve impulse travels down, and then finally the synaptic terminal, which releases the neurotransmitter glutamate. And one cone cell converges onto one bipolar cell, so it gives high visual acuity. So the photoreceptor cells turn the light energy into electrical signals, and these electrical signals transmit to the bipolar cells. The bipolar cells then process this information and send these signals to their axons. The impulses then pass through the ganglion cells to their axons and then transmit to the optic nerve, which then goes to the visual cortex in the brain. But how are these electrical impulses actually generated? So let's look at the process of phototransduction. First of all, just to get started with some key points of phototransduction. And phototransduction converts photons into electrical signals that are then transmitted to the brain. And this occurs on the light-sensitive photoreceptor cells, the rods and cones. And so photoreceptor cells actually hyperpolarize in response to light, and they reduce the amount of neurotransmitter that they release. And this is quite unique to photoreceptor cells, as most cells, when stimulated, depolarize and actually increase the amount of neurotransmitter they release. And there is 3 main stages to phototransduction. The first one is activation of the visual pigment rhodopsin. The second stage is a signal transduction cascade, which is a series of biochemical reactions. And we then have hyperpolarization of the photoreceptor cell and a reduction in neurotransmitter that is released from the synaptic terminal. So let's start with stage 1, activation of the visual pigment rhodopsin. So rhodopsin is the visual pigment, and it's a G protein coupled receptor. So that just means it's coupled to a protein called transducin, which is a G protein. Rhodopsin is activated by light, and you'll remember that it's formed from retinal and opsin. And in its inactive format, retinal and opsin are attached, so these 2 molecules are stuck together. But when light enters the photoreceptor cell, the rhodopsin molecule undergoes a conformational change in shape. So the retinal and opsin molecules change in shape, and this change in shape causes them to detach from one another and remain unbound. And now that retinal and opsin are unbound, the rhodopsin molecule is in its activated format, so it can signal to downstream molecules in order to generate the nerve impulse. So let's go on to stage 2 of phototransduction, the signal transduction cascade. Now this is quite a complicated process, as it involves a number of different molecules and signalling cascades. So we have our activated rhodopsin molecule, which is coupled to the G protein transducin. And when rhodopsin is activated, it also activates the G protein that it's coupled to. So GDP is converted to GTP on the alpha subunit of the transducin molecule. And when this happens, the transducin molecule actually leaves the rhodopsin. So it leaves the molecule and activates a different enzyme. And it activates the enzyme PDE. And when the enzyme PDE is activated, it triggers conversion of cyclic GMP to GMP. And in the photoreceptor cells, along the periphery, we have a number of cyclic GMP gated ion channels. When there's a high concentration of cyclic GMP within the cell, these ion channels remain open. So it allows sodium to enter the cell along its concentration gradient. When cyclic GMP is converted to GMP, the concentration of cyclic GMP reduces, so cyclic GMP can no longer keep the ion channels open, and these ion channels close, so the influx of sodium is inhibited, and this is how the cells hyperpolarize. So when the cells hyperpolarize, they reduce the amount of neurotransmitter that's released from their synaptic terminal. So let's go on to the final stage of phototransduction, hyperpolarization of the cell. And as I say, this is unique, as most neurons actually depolarize when they're stimulated. So when a neuron hyperpolarizes, its membrane potential actually reduces, it becomes more negative, and it goes from around minus 55 millivolts to around minus 70 millivolts. So in the light, sodium influx into the cell is inhibited, and this is what causes the hyperpolarization. And this reduces the amount of the neurotransmitter that is released from the synaptic terminal. But if glutamate is an excitatory neurotransmitter, how would a reduction in neurotransmitter stimulate the downstream neurons? And this is because the bipolar cells are quite unique, because they're one of the few cells that glutamate actually has an inhibitory action on. So if you reduce the amount of neurotransmitter that's released from the photoreceptor cells, it reduces this inhibitory action on the bipolar cells. So the bipolar cells generate a nerve impulse, they become excited, they generate a nerve impulse, and this is transmitted to the ganglion cells, which then goes to the brain, to the visual cortex. And that is the process of phototransduction. So finally, just some summary points of what we've covered today. So we've covered the anatomy of the eye, so the various structures within the eye and what they do. We've talked about the journey through the human eye and the different structures that light has to pass through in order to reach the retina, the photographic plate at the back of the eye. We've also talked about the retina, so it's a multi-layer structure of nerve cells which contain the photoreceptor cells, the rods and cones, which are the light sensing cells. And then we've covered the process of phototransduction in three main stages. So activation of the visual pigment rhodopsin, the signal transduction cascade, and finally hyperpolarization of the photoreceptor cell. So if you want to learn about this topic a bit more, here are some further resources that you can access. There's a number of good human physiology textbooks that explain the process of phototransduction in a bit more detail, so you just need to go to the relevant chapters in those human physiology textbooks. There's also a number of websites that can offer a good background but don't really give much detail, but they can be a good starting point to those who don't have much knowledge of the eye. There's also a few video tutorials that look at the process of phototransduction, and then also you can test your knowledge after you've done all that and see what you know about the eye using some online quizzes. And that brings me to the end of my talk today. I hope you've enjoyed it. Thank you very much for listening.